Welcome to this lecture on animal biotechnology. Today, we are going to explore a foundational technique known as calcium chloride transformation. If you have ever wondered how scientists genetically modify organisms or mass-produce life-saving medicines like insulin, it all starts right here. We will build your understanding from the ground up, so let us dive in. To understand how this technique works, imagine a microscopic fortress with thick, heavily guarded walls. This fortress represents our bacterial cell. Now, suppose you want to sneak a highly important instruction manual inside this fortress. In biotechnology, this instruction manual is a circular piece of DNA called a plasmid. This plasmid contains the new genes we want the bacteria to express. However, getting this DNA inside is not as simple as just walking through the front door. The cell has natural defenses that we must somehow bypass. This brings us to the major problem we face. Both the fortress walls, which are the bacterial cell membrane, and our instruction manual, the DNA, have a strong negative electrical charge. As you might know from basic physics, like charges repel. Just like two identical ends of a magnet pushing away from each other, the cell membrane and the DNA strongly repel one another. Because of this intense electrical repulsion, it is absolutely impossible for the DNA to just slip through the membrane naturally. We need a clever trick to overcome this barrier. Now that we understand the barrier, let us look at how scientists solve it. We treat the bacterial cells with a cold solution of calcium chloride. In this solution, the positively charged calcium ions act as a microscopic bridge. They sit between the negatively charged DNA backbone and the negatively charged phospholipid bilayer of the cell membrane, effectively neutralizing the repulsion. When the cells are treated this way, we call them competent. This means they are physically and chemically primed and ready to take up the extracellular DNA. The doors are unlocked, but not yet open. With the electrical charges neutralized, we move to the critical next step. We subject the competent cells to a brief and sudden increase in temperature, a process known as a heat shock. This sudden shift creates a thermal imbalance across the cell membrane. Because of this imbalance, temporary physical pores, or holes, are forced open in the bacterial fortress walls. The sudden flow of fluid sweeps the plasmid DNA right through these temporary pores and safely into the cell. Just like that, our secret message has successfully bypassed the guards. Let us visualize everything we have discussed so far in this diagram. On the left, you see the competent cell formation. Notice how the positively charged calcium ions are bridging the gap between the negatively charged plasmid DNA and the bacterial cell membrane. In the middle panel, the heat shock is applied, typically at 42 degrees Celsius. You can clearly see the temporary pores opening up, allowing the plasmid to enter. Finally, on the right, we have the recovery and plating stage, where the successfully transformed bacterial cell is allowed to grow into visible colonies on an agar Petri dish. Before we move on to the mathematics, let us understand why this matters in the real world. Without calcium chloride transformation, the entire modern field of genetic engineering would simply not exist. It is the reliable workhorse method that turns ordinary bacteria into microscopic manufacturing factories. For example, scientists use this exact technique to insert the human insulin gene into bacterial cells. This allows us to mass-produce life-saving insulin for diabetic patients globally. It is also absolutely essential for developing new recombinant vaccines and creating specialized enzymes for industrial use. We are literally rewriting biological software. Now that we know how the biological process works, we must ask, how do we know if our experiment was actually successful? In the laboratory, we need to verify if our competent cells were healthy and if our heat shock was timed perfectly. To do this, we calculate a standardized metric called transformation efficiency. This metric tells us exactly how many bacterial cells successfully took up the plasmid DNA for every single microgram of DNA that we originally put into the tube. It is a crucial quality control step in any biotechnology lab. This brings us to the mathematical representation of our experiment. The formula for transformation efficiency, or TE, is calculated by taking N divided by M multiplied by V total divided by V plated. Let us break down these symbols. N is the number of glowing colonies you physically count on your Petri dish. The variable M is the mass of the plasmid DNA you added, measured strictly in micrograms. V total is the total liquid volume of your recovery tube, and V plated is the tiny fraction of that liquid you actually spread onto the agar. If you increase the mass of DNA but get the same number of colonies, your efficiency decreases, meaning your cells were not very competent. Before we calculate anything, let us think about what this formula actually tells us physically. If the number of colonies, N, increases while everything else stays the same, your efficiency, TE, increases. This indicates a highly successful experiment where many cells took up the DNA. Conversely, if you use a larger mass of DNA, M, but get the exact same number of colonies, your overall efficiency, TE, decreases. This lower number suggests that your bacterial cells were not very competent, meaning they struggled to take up the genetic material despite having plenty of it available. Let us see this formula in action with a concrete example. Suppose you perform an experiment and count 250 colonies. You used 0.05 micrograms of DNA. Your total volume was 1,000 microliters, and you plated 100 microliters. First, we identify our variables and substitute them directly into our equation. We divide 250 by 0.05, which gives us a base value of 5,000. Then we divide our total volume of 1,000 by our plated volume of 100, giving a dilution factor of 10. Multiplying these gives our final answer. We successfully created 50,000 transformed cells per microgram of DNA, which is a very solid laboratory result. Now, let us look at a more advanced scenario involving a massive number of transformed cells. Here, you only used 0.01 micrograms of DNA, and you only plated 10 microliters from your 1,000 microliter tube. You count 300 colonies. We plug these new values into our formula. Dividing 300 by 0.01 gives a large base number of 30,000. Our volume correction factor is 1,000 divided by 10, which is 100. Multiplying 30,000 by 100 gives us 3 million transformants per microgram. This exceptionally high number proves that the calcium chloride treatment was incredibly effective at neutralizing the membrane charges. We have covered a lot of ground today, so let us review our key takeaways. First, we learned that calcium chloride is essential because its positive ions neutralize the repulsive negative charges between the plasmid DNA and the bacterial cell membrane. Second, the sudden heat shock is what actually creates the temporary physical pores, sweeping the DNA inside. Third, we can mathematically verify our success by calculating the transformation efficiency, which accounts for DNA mass and volume dilutions. You now understand the foundational technique that makes modern genetic engineering, from insulin to vaccines, entirely possible. Great job following along.